And thank you very much for the introduction. And good afternoon to everybody here. So as mentioned before, this is a presentation given about some work that came out of a collaboration in a European project called Positive, which officially finished beginning of the year, well, in February. And we're hoping, fingers crossed, to get paid by the end of the year. So principally, we'll be discussing, say, porous alumina, which we used. And we're using form birefringence in a polarimetric setup. For optical biosensing, we're using a copolymer to functionalize the surface of the pores. And we use quantum dots as signal enhancers. And we're showing some real immunoassay results. Okay, so it's not just about we've got a sensor and we measure some volumetric sensitivity, and we could do some biosensing, real biosensing, real microfluidics. Okay, we haven't used serum samples or blood samples, but everything done in buffer. So first of all, before we go any further, so work principally done by my student, who's now finished his PhD. He's now working in Germany, unfortunately, like a lot of Spanish researchers, and these colleagues from the various institutes and companies which the chair has introduced. So the work is performed in the unit for optoelectronic materials and devices, but since August I've now moved across the corridor. I'm now in this GES group, where I have pretty much an autonomous group. So the history of the group I lead is starting about 2008, and these are the projects that I've managed along the way, European projects. So we're finishing Positive and now in the Can-Do project. So these are the interests. We're looking to nanostructured materials for novel matter-light interaction control to provide added functionality. And we're looking to have these optical biosensors, which are both multiparametric and highly integrated. So we're looking at things of nanocomposites, both plasmonic and photonic, including nanoparticles, quantum dots, graphene platelets, and things like this. Optofluidics is very important as well, and SIRS, which is in the group I now belong. In terms of publications, in the projects, the first three projects have been over 120 papers, 30 peer review. From the group, there's been 9 publications since 2011, which is not bad because it has been pretty much me and one student, so it's and a lot of other work to do. And 36 conference papers, 9 invited, patent. And so the funding has all come from these projects, and I'm pulling in regularly about 150,000 euros per year from European funding, which leaves me about 120 once the university takes their cut, as it were. So because of these projects, I have a lot of collaboration and partners, a whole portfolio, both industrial and academic. So we're currently located at the University of Valencia, which I have to hop over a bit later for a meeting, which is why this has been brought forward. So apologies if that's disrupted your timetable at all. So before we go any further, just introduce the Can-Do project, a little bit of dissemination. So this is looking at pancreatic cancer, okay, and we're looking for CTC cells, detect them, count them, to give indication of how advanced your cancer is. So these are the various partners. I won't introduce them, but what we need here is reliable and sensitive cell sorting technology, and we're integrating established technologies to develop this diagnostic platform. We can isolate and characterize the CTCs, and if we can get early stage cancer diagnostics, and therefore we can follow on a very periodic basis the advancement, okay, of the cancer. Advancement or retreat, if you will, depending on how it responds to treatment. Anyway, back to the main point of the presentation. This project has just finished looking at allergies. Okay, allergies is what these. Oops, I shouldn't press that button. Allergies is the what they call the real millennium bug. Affects 1 in 4, and it's rising about 5% per year. Some 50 million US citizens, costing about $18 billion to the economy. And about 1 in 3 EU citizens, about 45 billion per year. And within that, food allergies, which is defined as sensitization to food products, they can have some very nasty reactions, even respiratory or cardiovascular problems, and affects about 8% of children, some 15 million Europeans, and it's an increase in prevalence. So we need something that detects them. And in fact, the actual IgE concentrations in the patients gives a good likelihood indication of relevant food allergies. So this is what we're looking for. We're trying to look at the concentration levels of IgE. Okay, and things like the toddlers in sensitization to hen's egg, it actually gives a good prognostic marker for future respiratory allergies. Okay, so there's lots of interesting information come out. This is how things are done currently. It looks a little bit medieval. They draw up the patient's back. This is for an adult. And then they scratch the skin with little extracts of allergy. Unfortunately, for a child who's 2 or 1, that can't be done because the back is too small. Sorry, it normally happens on the arm, but for an adult, for a child who's too small, they have to do it on the back. So they have to hold the child down, pretty much hold them down, and then for an hour or so or more, they're scratching the back. And as you can imagine, if you have parents here, it can be pretty traumatic. So, and it can also be dangerous as well because you can have a severe reaction. So we're looking for a suitable replacement test. Most of them are lab-based at the moment, blood tests. So you take an extraction in vitro. Point of care, there are a few, and they're limited. They're semi-quantitative, or they just test a few allergens, okay? So we're looking for something which is good sensitization down to 0.24 nanograms per milliliter, and tens or potentially hundreds of allergies in 15 minutes. So let's start looking at technology. So those are the unique selling points of technology which we would like to have in order to take it to market. So we created this device with a high surface to volume macroporous birefringent membranes, and you integrate them with interrogate them using phase locked loop measurements. And this is the Project Positive, and this is what Positive stands for. These are the consortium partners and the various partners. You can look at the slides, you can download them later. So, the porous materials. Why porous materials? Well, you have a very large surface area where you can bind your analytes to, your receptors and your bioanalytes. So using all of the bulk and not just the top surface as you do with most of the sensors. And in the same way, you can actually push the sample through the pores perpendicularly, and in that way, they can actually get to the surfaces very, very quickly because you're less than a diffusion length away. Okay? So you get greater sensitivity, potentially lower detection limits, and up to now, interferometry has been used. You see a paper here from Alvarez, nothing to do with the student that did this work, and you see a typical assay taking place. Problem being is because you have various different layers of different levels of porosity, normally they're closed off, so you can't push it through. So it takes normally so you need to have below 100 nanometers, which makes difficulty for fluidics. The porosity on top of the substrate, so the pores tend to block, or you have very slow hours, or I've seen even like 24 hours it takes to actually fill the pores. So it's not point of care, and it's certainly not real time. So we want to go beyond that. So this is what we do. As mentioned before, we have freestanding membranes. We started with poured silicon, and we moved on to alumina oxide. And we're using 200 nanometer pores or greater. And we use polarimetry so we have a 45-degree incident angle, which you can see here. Okay, and we see how the birefringence changes as you coat the surface or as you fill the pores. Okay, so that's the refractive index is the birefringence is directly related to the refractive index of the pores. So with this, you have a flow-through mechanism. You put target delivery for fast sensing responses, small sample volumes, which is very important because the reagents are very expensive, and fast assays. So we did this previously for an epoxysilane functionalized surface. This was published last year with proceedings. Now we're showing a different surface chemistry and different immunoassays. So we use this functional copolymer, which has demonstrated lower fluorescence, less non-specific binding, therefore greater specificity, and you mobilize the allergens. They're more stable over a longer time. So this is very important, of course. We're using off-the-shelf Wattman filters, okay? So they're not fantastic, but they're cheap, they're available, and we've got ahead. You can buy some great tailor-made membranes, if you like, from a company called Smart Membranes. I've nothing to do with them. I'm not advertising, but those are the ones who they do a two-step electrochemical etch, and certainly that's what we would like to use going forward. So following the functionalization and the immobilization, we transfer the membranes onto support chips, mechanical support chips, and we use PMMA as an adhesive to stick the membrane onto the chip. Then we mount them in the flow cell, and in order to measure reproducibility, we did a whole series of bulk refractive index experiments. So we did 15 minutes of buffer, so that was PBS in tween, and then after a few minutes, we introduced various concentrations of salt. Okay, so you can see it's 0.2%, 0.4%, 1%, etc. And then if we plot the various changes in phase measured, get a change of the refractive index due to the salt, then we get this sensitivity, okay, 5.2 radians per refractive index unit. And this comes out to being 5 times 10 to the minus 6 refractive units, and we have a system resolution of about 2.7 times 10 to the minus 5 radians. And this sensitivity actually compares quite well to the theoretical, which is calculated from Brueggemann model. So now to the Amino Sensing, which we did afterwards. So this is what we have. These are our pores. We're looking down this way into the pores, and the flow will go this way. We have here on the surface so it's functionalized first of all. Then you have the beta-lactoglobulin. Okay, it's not quite the same size as the key that are here. On top you have the primary antibody, secondary antibody, and then you have what we call a signal enhancer, which is a strep-cavity-encoded quantum dot on a bead. So we purged the cell first of all. We introduced between 15 minutes. The baseline showed good stability, so that's when we started started mounting the assay, if you will. So we introduced, as I mentioned before, the first antibody, 10 minutes, run the buffer of 6 minutes, then the secondary antibody, then 10 minutes, and then we introduced the quantum dots at 2.5 nanomolar for our signal enhancers. The polyclonal secondary antibody shows a larger response than the primary. I mean, this data's not here, and the quantum dot saturated captured secondary antibodies. Okay, and we saw 5 times enhancement of signal for the quantum dots adding than just over the secondary antibody response. So what we did after that is then vary the concentration of the first antibody, okay, and then see how the response comes when we add the secondary and the quantum dot. As you can see here, as we start off with the primary antibody, the larger the concentration, the bigger the response you get. Okay, here we cut off the flow, but as you can see, it's considerable. So we plot this then primary concentration against the actual phase retardation change, okay, the phase change measured, and from that, so we did this for 12 different membranes. It's not all on the same membrane, and from that we got a response, and the limit of detection we took as the analyte concentration that can be reliably distinguished from the noise. Okay, and I don't know if you've seen this before. We took this out of quite an obscure paper, but the limit of detection is defined as the limit of blank plus 1.645 times the standard deviation of the lowest concentration sample. Okay, so the lowest concentration. And the limit blank is defined as the highest analyte concentration value expected to be obtained for a sample containing no analytes. And then from that, this is your limit blank, that's defined. And working all this through, we came out with a limit detection of 33.7 nanograms per milliliter, which is a little bit disappointing, but actually most of the restrictions of that came from the actual mechanical setup, which was really kind of ad hoc. It wasn't really done repeatable, and the actual sort of cell holder and the support could be done a lot better. And so we reckon we could get that down close to what is the actual signal noise level, which is just the it's the sort of standard deviation of well, this is the noise, the level on the noise, and that's 3.7 nanograms per milliliter. And in fact, a lot of people actually quote that as the limit of detection, which is not strictly. Okay, but we should, in principle, this could be got down to about here, without changing any of the measurement scheme. So if we compare these to flow over porous membrane sensors, which I showed, I think it was on the 4th slide, which is commonly done to now. Michael Saylor's done a lot of work on this. We see a more efficient convective delivery of the analytes. We see an assay time of less than an hour compared to 5 hours or more. We have 1 milliliter of 1 microgram per milliliter primary antibody, okay, in 10 minutes, rather than using 50 milliliters of 0.1 milligrams per milliliter in 100 minutes. Okay, so considerably less reagent and therefore a lot cheaper the assay, which is where most of the cost goes in when you actually work it out in the healthcare for your measurement. So move on to conclusions. I don't know if I'm on time or, yep, okay. So what we've done is we've used this novel procedure to coat aluminum oxide that hasn't been done before with copolymer, or the copolymer being used on other surfaces, where we demonstrate because we've seen less non-specific binding and more stability for mobilized allergens than on epoxysilane coated membranes from a previously published work. The bulk refractive index measurements, they showed a detection limit of 5 times 10 to the minus 6 refractive index units, which is kind of more or less par for the course. It's not state of the art, but a lot of values come in there. Anything less than that is not very attractive. But we got a standard deviation less than 5%, okay, from one membrane to the next, one measurement to the next, okay. So that's very important when it comes down. If you want to take this to the market, if you want to create a technology, that is very important, okay. You want very little differences between using one chip and the next, okay, for exactly the same measurement. In terms of stability, the functionality of the coating and spotting procedures, we evaluated those, and we looked at the binding between the allergen protein and its cognate antibodies in real time. And from this, we saw a 5 times signal amplification for the quantum dot over just the secondary. The noise floor was 3.7 nanograms per milliliter. Okay, the limit detection was a bit higher than that, but we could get down to that in principle. And the total assay time was less than an hour. And compared to flow over, we get 5 time assay time reduction, 3 time reduction in the analyte consumption. And this is all a route going towards rapid, low-cost, real-time optofluidic biosensors for small and therefore less costly sample volumes. So I think that's just to say thank you, and thank you for my colleagues.